Subject for today and Wednesday are the Archaea. And I know you guys have heard about these organisms before. It used to be that I had to describe in great detail what these things were, and there were students who'd never heard of them, and so forth. That's not true anymore. These are organisms that, when I was taking general micro, were thought of as just weird bacteria. Things like Halobacterium were thought to be an extremophilic Pseudomonas. Sulfolobus was thought to be related maybe to things like, the Actinobacteria, things like, Arthropacter or Corynebacter. Of course, nothing could be further from the truth. I actually was taking general micro the year this was discovered. It made the cover of the New York Times. New life forms discovered. They weren't discovered. It's just that we realized that they were different than bacteria. The instructor of the class that I had didn't understand at all what this discovery was about. As the students left the classroom thinking that we discovered silicone life forms on Earth or something like that. There's nothing like that. But they are a third kind of living thing on Earth, with a common ancestry with the eukaryotes and bacteria, and specifically with the eukaryotes. Although in many ways these things are like bacteria in terms of their gross phenotype, when you look inside at their works, very often they are like eukaryotes instead. And this is one of the reasons why people like to study them, is because they provide a simple system without all the complexities that eukaryotes use, but where the fundamental machinery is eukaryotic-like. There are two major branches of Archaea, the Euryarchaea and the Crenarchaea. And this is color-coded based on phenotype. The red ones are halophiles, organisms that live in soda lakes and saturated brines. The greens are the methanogens, organisms, extremely anaerobic creatures that make methane for a living. And the yellow ones, which are sulfur metabolizers. They oxidize, reduce, or in one way or another use sulfur as a primary form of metabolism. I know that's a kind of a generic phenotype, but we'll see what that means. There are a couple of organisms like Nanoarchaeum and Corarchaeum and some other things that have been recently discovered that may be related to these other main branches or may not. The nature of that is unclear. It's looking like Nanoarchaeum is just a Euryarchaeote. Corarchaeum is probably a Crenarchaeote, but there's some newer ones where that's a little unclear. If you want a general rule for Archaea in terms of how they compare to bacteria and eukaryotes, that general rule might be that their phenotype and their metabolism is bacterial-like, but their machinery is eukaryotic-like. And so things like Dna Polymerase, RNA polymerase, the transcription apparatus, glandular motor systems, etc., are basically eukaryotic-like. Glandular motor systems, in terms of their enzymology and cell structure, they're generally bacterial-like. And in a few ways, they're different than either one. Morphologically, they look like bacteria. They're mostly small, in the few micron range. They come in the same kind of shapes bacteria do, because those shapes are defined by the physics of living at that size, right? Archaea generally reproduce by fission or by budding. The cell envelope is one of the ways in which Archaea are different than both bacteria and eukaryotes, and particularly in the lipids. Well, first of all, they don't have peptidoglycan. Bacteria have peptidoglycan in the cell wall for the most part. Archaea do not. None of them. Some of them have structures that are similar to peptidoglycan in that they are polysaccharide chains cross-linked with amino acids, but they're clearly not homologous. The lipids, though, are really interesting because both bacteria and eukaryotes have phospholipids, right? And those phospholipids are esters between glycerol and long-chain fatty acids. Archaea do not have this. They have glycerol phosphate with a head group on it, just like bacteria and eukaryotes do. But the side chains are alcohols, not acids. And the result is, when you bring those together, that's not an esterification, that's an, I don't even know what you call it, etherification. You end up with an ether linkage instead of an ester linkage. It would have to be a double bond O here, right, if it were an ester. And the chains aren't more or less linear chains. So when you think about bacteria and eukaryotes, those chains are linear, whereas and they're linear because of the way they're made, right? It's acetate, just polymerized. These things are isoprene polymerized. So they're synthesized in a completely different way. The synthesis of these things is related to the photopigments of plants. That's the kind of chemistry that they use. So think of this as like phytan. And so they're, every, 1, 2, 3, 4 carbons, there's a methyl group that sticks out. In bacteria and in eukaryotes, where you have double bonds, they are interrupted. So here are 2 double bonds, 2 unsaturations, and they're interrupted by a methyl group. What that means is that the 2 double bonds are independent. They're not resonant with each other, right? In Archaea, I don't think they show a picture of it, but where there are unsaturations, they typically are not separated by a methyl group. And the result is they are resonant with each other. They're conjugated double bonds. And the result is they're rigid. And you can't rotate them easily. They're usually cis rather than trans, and so it puts a big kink in the chain. They have these ring structures embedded in the lipids. The really interesting thing, though, is that in both eukaryotes and bacteria, these things are, about 18, 20 in length on average. They vary a little bit beyond that. And you have 2 of them, one on each side of the membrane. These are lipid bilayers. Archaea do not have lipid bilayers. They have lipid monolayers. And so some of them are like that, but many of them are 40 carbon chains that stretch all the way across the membrane. This has a profound effect on the fluidity of this membrane. And this picture is not accurate in the sense that this shows 2 chains here and they both connect to the same glycerol over here, right? But that's not the way it is. This chain will be attached to an arbitrary glycerol. There's no decision that it has to be the same one. And so you can imagine this could be connected to another one, and that one to a different one. And you end up with this giant 3-dimensional network of lipids. Think about what that does to the fluidity of that membrane. Why would you do that? What if you live at 115 degrees centigrade? This has the effect of allowing you to grow at those kinds of temperatures. And like bacteria, they tune their membranes all the time to maintain the optimal fluidity for that temperature. This also means, though, that getting stuff in and out of Archaea is fundamentally different than it is in bacteria. And there are lots of organisms, lots of Archaea that can't even take up glucose. They don't have the pumps for it. That membrane is the only membrane they have. They have a single cell membrane. There's no outer membrane. They don't have peptidoglycan. I mentioned that. They very often do have glycoproteins that are cross-linked to add rigidity to the cell, or an S layer, a semi-crystalline protein coat on the outside of the membrane. One of the things that's come up recently that's really interesting is the business of their flagellum motor system. Flagella in bacteria look like flagella in Archaea, but they are fundamentally different. They are not homologous. Like in bacteria, there's a motor complex and a fiber that's bent. The helix runs the opposite direction, by the way. But they are based on a different motor complex. And so these motor complexes are homologous to secretion systems. And that cranking motion is used to pump things in and out, or in the case of some virulence factors, to drive basically a syringe needle into a host cell as a way of injecting stuff into the organism that become a pathogen. The flagellar motor complex in bacteria, of course, is proton-driven. It's driven by the proton gradient. The fiber is hollow, and it's built from the base out, right? The protein subunits go along the length, and they stick to the end. In Archaea, again, this is fundamentally different. It's based on a different secretion mechanism. I can't remember which. Type 3, oh, here we go. Type 2 secretion system. It's related to type 4 pili. This is an ATP-driven motor. It's driven directly by ATP hydrolysis. No proton gradient required. The protein is built from the inside out, and so proteins are added here and pushed out the filament. So they're more like pili. And obviously the motor complex is different because it's not sitting in a cell wall, outer membrane, structural sort of context, where you've got a bunch of plates, one for each of the layers of the motor. These things have ribosomes that look like a little bit like bacteria and a little bit like Archaea. In terms of size and function, they look more bacterial-like. They bind to Shine-Dalgarno sequences, for example. But if you start mixing and matching pieces, taking a protein from sulfolobus and building it into ribosomes of other organisms, the components are more interchangeable with eukaryotic ribosomes. Transcription. These things have a single RNA polymerase, like bacteria, but that RNA polymerase is a dead ringer for RNA pol II in eukaryotes, not bacteria. Promoters are tautoboxes, and tautobinding protein binds to it, and that's how transcription is initiated, just like in RNA pol II genes, mRNAs, in eukaryotes. And they're sensitive to the same inhibitors and all that kind of stuff. The genomes are generally circular, as is true generally in bacteria. There's not a specific terminus of replication, and very often there's more than one origin of replication, like in eukaryotes, right? These things are generally positively supercoiled, especially the thermophiles. And so bacteria negatively supercoil the DNA, so eukaryotes, that makes it easier to pull strands apart, right? So you can put proteins in and do all the stuff you need to do. These organisms, particularly the thermophiles, positively supercoil the DNA to make it harder for the two strands to come apart. Again, very useful when you're growing at high temperatures. The DNA polymerases are eukaryotic type DNA polymerases. And the list goes on and on. You can look at these things, and most of the time, any single property you look at will look like either eukaryotes or bacteria. But you don't usually know which it's going to be like. Assuming that something is going to be bacterial-like will get you into trouble. Occasionally, there are some properties that are unique to Archaea and are different from either eukaryotes or bacteria. Not many, but some.